In this tutorial, I'm going to show you how to perform RNA Seq analysis using your browser. So what we are going to do is to use the GEO2R environment to perform the RNA Seq analysis. So let's get started. So first of all, you need to visit the GEO2R page. And this is the page. I'll leave the link to this page in the description box. Alright, so we are on the page that allows us to analyze RNA Seq datasets that have been made available on the GEO database, that's Gene Expression Omnibus. So I'll leave the link to this page in the description box. So when you are here, you have to specify the accession ID of that study that you are interested in. All right. Now, let's just say we don't have a study yet. We can search for the study that we want and then get the accession ID and then paste it here. So for that, we have to go to the GEO dataset page itself, which is here. I'll leave the link to this page in the description box. So when we are here, you have to search by supplying some terms here or keywords, and then you have hits and you can select the appropriate study. All right. So let's just say we are interested in studies that have been done for zebrafish. All right. So in this tutorial, because we are going to analyze RNA Seq data, we are going to take RNA Seq studies that were done using zebrafish samples. And so I will type zebrafish here, and then I am going to search. So when I search, I'll get the hit here. Now we have lots of hits being returned. We have over 40,000 here. That's a lot. And so ideally what you want to do is to be as specific as possible so that you have fewer lists displayed and you can select the one that you want. And so we can add something else. So let's just say we are interested in cancer studies that have been done using RNA Seq samples from zebrafish. Then we can add the term cancer or tumor, whatever you want, or whatever term that is of interest to you. So to do that, you have to go to the advanced. All right. So you don't even need to have the zebrafish typed here. You can just clear it. You have to go to advanced. So click on advanced, and then when you click on advanced, you have another page being displayed for. So here you can enter your terms or keywords. So I'll start with cancer. I'm interested in cancer studies. And then here you can also select the field which has that term. So all fields, attributes, attribute name, etc. So you have to decide what you want, but I'll just leave it as this, right? But later on you can modify to see the result that come up. And the second entry box here, I'll also use zebrafish. Right, you can also add more, so that one depends on you, but I'll just leave it like this. So once you have entered your terms and you are okay, you can click on search here. So click on search, and then when you click on search, you have some hits returned for you. So you'll notice now we now have 2,339. Right, there has been a drastic reduction in the hits that have been returned. So here you can scroll and then look at the study or studies of your choice. Now for studies that can be analyzed with GEO, you will find this text here, analyze with GEO there. So when you find it there, then you can click on this to analyze. So let's just scroll down to search for other studies. We are going to use one example here. So in this tutorial, we are going to use this one. So let's assume that we are interested in this study. Mine is number 13. All right. So you have to click on the Analyze with GEO, or you can also just take a note of the accession ID. I'll just copy it for the meantime. Right. So you can click on Analyze with GEO, and then you'll have it displayed for you like this. But if let's say you already know the accession ID, like in this case, then you can just come to the GEO2R page itself here, and then you can paste the accession ID. Right. I'm going to leave this accession ID in the description box. Or you can also try with any accession ID of your choice. That's fine. So after pasting it here, you can come on Sets. Come to this side here, Sets, this button, and then click it. So when you click it, then you have the data sets being displayed for you. So this is how we start the GEO2R process. All right, so let's continue. Okay, so now we can proceed. Now, if I want to analyze RNA Seq data, then one of the things that you will do is to identify genes that are differentially expressed across the experimental conditions. So that means that we need to identify the groups that we use, and then we can proceed with the exercise. If you take a look at this table here, just look at the column treatment, this column here, treatment. We see two groups. We have control, we have TGF-beta. So these are the groups that we will use. So before we do analysis, we need to assign the samples to their respective groups, i.e. control and then TGF-beta. All right, so to do that, we will need to use the define groups button here. Now we will come back to this one. All right, if you are going to define the groups, it should be in a certain order. All right, so let's take a look at an example activity here. Let me just open this link and then check it here. I'll leave the link to this page in the description box. So when you are here, come to assign samples to each group. Let's click it. So when you click it, you will see an example here. So here we see that the control group is to be defined last. Okay, it should be the last group to be defined. So I'm going to follow the same instruction here, the same step that has been indicated here. I repeat, I'm going to follow the same steps that have been shown here. Now let's get back to our GEO2R page. So here we have control, we have TGF-beta. So based on what I have seen here, all right, we will define the TGF-beta first, and then we'll come to control. So I'll come to define groups, and then I'll click it. Then when I click it, I'll enter the name here in this entry box. So that will be TGF-beta. I'll use lowercase, and then I'll hit the enter button. So I'll have it created. Now let's create the other group, that is control. So that will be control. I hit the enter button or the enter key on my keyboard, and then that's also created. All right, so we have done that nicely. So now we have to assign the samples to these groups. All right, so to do that, it's very simple. You have to select the samples of interest, and then you click the group that they're supposed to be assigned to. So I will start with TGF-beta. So I have them here, all right? So I'll select, this is one of them, and then I'll also select this one. So with the selection, you just click it, and then you can click the other one. But if you click one and you click the other, you will see that the color has moved to the one that you clicked on. All right, so if you are selecting more than one sample, then you have to also use the control key. All right, so let's say I have selected this one. Now to select the other one, which is this one here, I'll press the control key and then I'll click it. So you'll see that both have been selected. So when a sample is selected, the highlight color will be this one here. It's orange or yellow or something like that. So, but you get the point. So you will see that the sample has been given this color. So now we have two samples selected. So let's select the other one, this one. Then let's select the other ones too. We have this, this, and then this. So we have all of them. So now you can click on the group, that is TGF-beta. So click it. And when you click it, you will see that the number of samples that you selected will be displayed here. And you also see that the color will change. I mean, the highlight color for those samples will change. So now we see this color. Now let's select the samples for the control group as well. So I'll click. I am still pressing the control button. So I'll just be clicking to select. And when I'm done, I'll come to control and then I'll click it. So this will also assign those samples to that group. So you see that the color for those samples have also changed. So now we have two colors here indicating two groups. All right, so that's what you need to know. Now, if you click on any of the samples, even though you have assigned them to groups, you still have this color coming up. So this color simply means that that particular sample has been highlighted, or you have selected it, or you have clicked it. All right, that's the point of focus now. So when you click on a different sample, then the color that was assigned to based on each group will be displayed again. Okay, so you don't have to worry if you see this color coming up. So just click on another sample, and then if indeed that sample was assigned to a group, then the color that it was used to, then the color that it was assigned to will be brought back. That's what you need to know. All right. So now let's proceed. So let's continue with the analysis. So now we have 6 samples for TGF-beta. We have 6 samples for control. So we can proceed. So you can hide the defined groups. So you can click this to hide it, but still your selections are active. So don't worry about it. So after doing your selections, after assigning the samples to their respective groups, then you have to scroll down to the bottom page, the bottom part of the page. And then when you are here, click on the Analyze button here, this one here. So click it, and when you click it, you will see this notification processing. So that means that the analysis is being done. So what we have to do is to wait for everything to get done for us, and then we can proceed. So let's wait for everything to get done for us. Okay, the analysis has been completed. So you will have your plots being displayed for you, and you're also going to have the differentially expressed genes being displayed for you. So you have this table, right, and then you can proceed with the rest of the activities. So let's take a look at some stuff here. So with the analysis, it's possible to change the options that were used. So when you are here, just click on options, and then you can select the options of choice of your choice. All right, so that one you have to do that, and then you can proceed. Another thing you can also do is to do the profile graph. So here you can specify a gene of interest, and then you have some information displayed for you. So let's try out an example. Let's get back to the GEO2R page itself where we have the results. Let's take a look at this table here. Let's take a look at this gene here. Let's say CTPS1. Let's get back to profile graph. Say it's CTPS1. And then let's click on set. All right. So when you click on set, you have the information being displayed for you. And you even take a look at this one. It says this tab allows you to view a specific gene expression profile graph by entering a gene symbol or some stuff here. So it displays the TPM normalized expression values across the samples. All right. So if you take a look at the gene here, you see the normalized values here. So we see that this gene is upregulated because it's highly expressed. Okay, so compared to the control, right? So this is also indicated in the table here. So if you take a look at this table here, right, you will see the log2 fold change 1.23. All right, so you also see the p-adjusted value, it's 0.00. Okay, so that means it's significantly expressed because the p-adjusted value is 0. All right, so we see that here 1.23. So it's upregulated basically. You can also try with the others. I mean, those genes that of interest to you. You can look at the top 10 or top 20, or maybe the first one here. We look at the first one, the most significantly expressed. That's what we have here. And then that is what we looked at here. So you can use this to get more information or to zoom into particular genes of interest. That's something you can also do here. But you can also click on the sample values to see the values here. So that's up to you. All right. Now, if you want the R script itself, let's close this. If you want the R scripts, then you can click on this one here. So this will generate the R script. So this R script, you can run it locally on your PC to also generate the results here. All right. So here you can just copy, let's say Control A, copy, and then paste to an R environment and then run it. So that's something you can also do. So you don't need to necessarily come to this page all the time. So if you know the Accession ID, you have an internet connection, then you can even change some of this information here, and then you can get your analysis done, right? You can also do this if you have your own data sets too. Then you can just look at those parts of the codes that can be modified. For example, you have your own data sets, you've done your differential expression analysis, then you can use some of the codes here to generate your plots. All right, so that's something you can also do. So This can help you get started when it comes to RNA Seq analysis. So that's what you need to know. So that is it. So you can also come back to GEO2R, and you can also download the plots here. So to download, you can just click it. You can click on explore and download like this. So you will see the figure here, like this. The figure here, let's just check it here. You can click on download significant genes. There's a CSV file. You can cancel it. And then with the imaging itself, you can right click. Let's go to this other one here. I'll do that here. So to download, let me just close this. So to download, you can right click, then go to save images. So this will save it for you. All right, so when I click on save images, I can just save it. All right, so I'll just type DEG differential express genes. I'll create a folder and then I'll just paste it there. So this is volcano plots. So I can click and save it, and it's done. You can do same for the others as well. Then you can later on explore them. It's important to explore. So we have all these cool plots that you can use. You can also download this table here. So you click on download full table. And then after clicking it, you have this coming up. So all I have to do is to wait a bit. And once everything is ready, you'll be given the file. So you can save that file. So you can save it wherever you want to save. I'll just save it here like this. And then I'll open it. So I can open the folder which has the data, and that is this one. So what I'll do is to open it with my spreadsheet software. So I'll just open it to see what I have. So I have all of them here. So we have your gene, PISF value, P value, etc. So this is the output for DESeq2. All right, so here you have them here. And you can also perform downstream analysis. For example, gene ontology. You can also look at pathway analysis, etc. So these are things that I'm going to cover in other tutorials. So once I'm done with that, you can check the description box. You'll find links to those videos. All right, so that's how we do it. So we can close it and get back to our browser. Let's come back here. And so if you're interested in more RNA Seq data analysis, then I will encourage you to check this playlist right here. If you're also interested in learning how you can use the Galaxy platform to run DESeq2 and perform your gene expression analysis, then you can check the video right here.